Welcome to our colloquium talk for today. We are especially pleased to have our own Professor David Gerard as our speaker. David completed bachelor's degrees in mathematics and molecular genetics and a master's degree in statistics at the Ohio State University before obtaining his PhD in statistics at the University of Washington, where his dissertation concerned the multivariate analysis of tensor formatted data sets. He then worked on a postdoc scholarship at the Department of Human Genetics at the University of Chicago, where he developed methods to account for unwanted variation in the analysis of RNA sequential data. While at American, he has since transitioned to genomic analysis of polyploid organisms, where he has found some success, publishing in several of the top journals, including Genetics and Heredity, where notably his works were selected as highlighted articles in their respective issues. Congratulations, David. Also, congratulations to David are that he recently received funding for his new work on polyploids from the National Science Foundation under a grant titled A Ustatistic Approach to Population Genetics. And on a personal note, I was the department chair when we hired David. And I thought that search committee did spectacular work, brought in several amazing candidates. And I was really happy that David was the person that the committee put forward. And then I did my very best to get David the best possible welcome package and get his salary set up well with with then Dean Starr. I guess I did a good job because David's here, and I will certainly say he's a wonderful member of our department. He's an excellent teacher. I always see students in his office working with him. And I think his research work is spectacular. And today he's going to be telling us about double reduction estimation and equilibrium tests in natural autopolyploid populations. So I hope I got all that correct.
That's all right.
Yeah. I will turn the floor over to David, and welcome.
Thank you so much, Steve, for the kind introduction. Let me share my screen. All right. Yeah, so today I'm going to be talking about a new preprint on equilibrium testing in natural autopolyploid populations. And this also concerns double reduction estimation. So first, I'll talk about what Hardy-Weinberg equilibrium is in diploids and how classical methods to test for Hardy-Weinberg equilibrium don't work when we look at polyploids. So diploids are organisms with 2 copies of their genome, and polyploids are organisms with more than 2 copies. Humans are diploid, but a lot of important plant species like blueberries and potatoes, they're polyploid. And the research for equilibrium tests is much more limited when it comes to polyploid organisms. So the issues that we encounter when we move from diploids to polyploids are caused by special occurrences in meiosis, so how gametes are formed. So there's some phenomena in meiosis that affect the methods that make the methods that are used in diploids not applicable when we move to polyploids. So I'll talk about 2 ways that we handle this: a likelihood approach and a new Ustatistic approach, which I think that I'm very excited about. So genomic researchers are interested in your genome, which is your DNA. It's made up of long sequences of these nucleotides, ATGCs. So at any location on the genome in a species, it's going to be the same from individual to individual. The differences, the few locations where there are differences, those are the interesting locations. They allow us to study what parts of the genome are responsible for certain traits. They allow us to explore the way that species have evolved or are currently evolving. These differences are very important. So a common type of difference in the genome is called a SNP, SNP. It's pronounced SNP, for single nucleotide polymorphism. It's just where a single nucleotide is different from individual to individual. So it's a location on the genome where a single nucleotide varies across the population. So if this is a small segment of a diploid individual's genome, so diploid individual has 2 copies of their genome. On one copy, they have ATT-CAA, and on the other copy, they have ATT-GAA. Then the SNP is the location where there's this difference, a C and G. And this individual has 1 C and 1 G. But other individuals at the same location on the genome might have 2 Cs or 2 Gs. So biologists have a special word for difference. They call it an allele. So in this, at this location, this SNP has 2 alleles. It has a C allele and a G allele. And typically, SNPs only have 2 alleles. They're biallelic. The reason is that SNPs are a result of mutation historically, and mutation is a rare phenomenon. And so it's very unlikely for there to be multiple mutations at the same SNP. And so usually at a SNP, there's only 2 alleles, one of the nucleotides and then the other nucleotide. And in this SNP, there's the C allele and the G allele. So how many copies of an allele an individual has, that's called their genotype, or their dosage. People use both terms. So the dosage for the C allele is the number of Cs that this individual has. It has 0 if an individual has 2 Gs and no Cs. The dosage is 1 if an individual has 1 C. And then the dosage is 2 if an individual has 2 Cs. So if we have a population of individuals, some of them have GG, some of them have CG, and some of them have CC, we're often interested in how these genotypes vary throughout this population. So if this is our population of individuals, we count up how many individuals have G, how many individuals have CG, and how many individuals have C. And we calculate the frequencies for these different genotypes. These are called the genotype frequencies. So there's 7 individuals with GG out of 20. So the genotype frequency for a dosage of 0 Cs, is 0.35. Now, another quantity of interest for biologists is the allele frequency. So the allele frequency is where you kind of ignore what alleles belong to which individuals, and then you just count up how many alleles of each allele are in the population. So here there are 24 G alleles and 16 C alleles. And so the allele frequency for C would be 16 over 40 or 0.4. All right, so these are 2 concepts for how genotypes vary in a population in terms of genotype frequencies, and how alleles vary in a population in terms of allele frequencies. So what Hardy-Weinberg equilibrium is it's a state of the population where if certain conditions are met, then we can represent the genotype frequencies in terms of the allele frequencies. And we represent it using these proportions. So PC, this is the allele frequency of the C allele. And if Hardy-Weinberg equilibrium is met in this population, then the proportion of individuals with the dosage of 0 is 1 minus PC squared. The proportion of individuals with the dosage of 1 is 2 times PC times 1 minus PC. And the proportion of individuals with the dosage of 2 is PC squared. So Hardy-Weinberg equilibrium doesn't always occur, but when it occurs, then we can represent genotype frequencies in terms of the allele frequencies using these equations. Now, this might look familiar to a lot of you. This just looks like the binomial proportions. It just looks like the binomial distribution. So if xi is the dosage of individual I, so the number of C's that an individual I has, either 0, 1, or 2, then Hardy-Weinberg equilibrium states that xi is going to be binomially distributed with size 2 and success probability PC, where PC is the allele frequency of the C allele. So in order for these proportions to pop up, you need to have the usual assumptions of the binomial distribution. So you need to have a large population size that you're randomly drawing an individual from, infinite population size, but large is good enough. And then the alleles of an individual need to be randomly sampled from the population. So the 2 alleles that each individual has is a random sample from the population. In order for this to occur, you need to have a lot of conditions on the population. So you need to have random mating, so each individual is equally likely to mate with any other individual. You need to have no evolutionary selection. So one allele isn't more advantageous, evolutionarily speaking, than any other allele. There's no mutation, so mutation is at a low enough rate that you don't expect there to be new mutations at that locus in subsequent generations. There's no migration, so there's no external population where there's migration to or from your reference population, and so on. So there's a lot of assumptions that need to occur in order for Hardy-Weinberg equilibrium to be fulfilled. But it turns out that in a lot of settings, in a lot of organisms, and a lot of populations, and in a lot of SNPs, Hardy-Weinberg equilibrium is mostly satisfied, that these assumptions are mostly satisfied so that Hardy-Weinberg equilibrium, we see the Hardy-Weinberg equilibrium proportions. So because of this, Hardy-Weinberg equilibrium is a common assumption in a lot of population genetic models. And if you have violations in Hardy-Weinberg equilibrium, the most common explanation for that is that there's a genotyping error. So that means that your biological assay that you used in order to read what sequences are on the DNA of some individual, that assay is corrupted at that location. So this is the most common usage of tests for Hardy-Weinberg equilibrium, is to detect loci where there's genotyping errors. So being able to detect violations from Hardy-Weinberg equilibrium is very important, because you want to include only SNPs or loci that satisfy Hardy-Weinberg equilibrium in order to apply more complicated population genetic models. So let's talk about the classical way that people do this in the case of diploid individuals. So the classical way is to just run a chi-squared test, right? So if we have counts of each of the different genotypes, so 3 individuals with C, 10 with CG, and 7 with GG, then we can calculate the allele frequency. We can estimate the allele frequency based off of the sample. So 3 individuals contribute 2 Cs, and 10 individuals contribute 1 C. So that's 6, 2 times 3 plus 10 to get 16, and 16 over 40 number of alleles, you get an estimated allele frequency of 0.4. All right. And then for the chi-squared test, you take your observed and you compare it against the expected count if Hardy-Weinberg equilibrium were fulfilled, right? So the expected count would be the number of individuals in your sample, here 20, times the expected, times the proportion that you would see if Hardy-Weinberg equilibrium were fulfilled, so 0.4 squared. For CG, 20 times 2 times 0.4 times 1 minus 0.4. And then for GG, 20 times 1 minus 0.4 squared. So these are the expected counts, and then you do the usual chi-squared test. So observed minus expected squared over expected. You compare it to a chi-squared distribution with 1 degree of freedom. 1 degree because there's 2 parameters under the alternative, where each of these genotype frequencies are allowed to vary except for the constraint that they sum to 1. Minus 1 parameter under the null, where the only parameter that you use is the allele frequency. So this is the classical way. You just compare your observed counts to the counts that you would expect under the binomial distribution. Now this is a really simple problem to explain, right? You're just comparing counts to whether or not these counts follow a binomial distribution. So it's surprising about how much literature there is on testing for Hardy-Weinberg equilibrium. The reason why there's so much literature is that this is such a fundamental state. This is such a fundamental thing in population genetics that it's very important to detect Hardy-Weinberg or violations from Hardy-Weinberg equilibrium under different scenarios. So people have expanded the literature to include, likelihood ratio tests, exact tests for cases when you have a small sample size or a rare allele, Bayesian methods, tests for multi-allelic loci, so where there's not just 2 alleles, but multiple alleles for different types of differences in the genome. If you have sex-linked loci, so loci on the X chromosome or the Y chromosome, you need to have different types of procedures in place in order to detect violations from Hardy-Weinberg equilibrium. Population structure, so systematic deviations in the genome, in the genotypes of individuals. This can cause deviations away from Hardy-Weinberg equilibrium. And so people have come up with ways to account for population structure when testing for Hardy-Weinberg equilibrium. And there's tons of literature on tests for Hardy-Weinberg equilibrium, and tons more come out every year. So this is my contribution to this field, is extending these tests to the case of polyploids. So polyploids are individuals with more than 2 copies of their genome. And there's different kinds of polyploids. So I'm going to talk about autopolyploids, which is where the genomes, all of the genomes are kind of more similar to each other. There are other kinds of polyploids, allopolyploids, where the genomes can be subdivided into subgenomes, where the subgenomes are more similar to each other than between subgenomes. So I'm just going to talk about autopolyploids. And examples of autopolyploids would be like potatoes or sweet potatoes or blueberries. So this is for polyploids. If we have a tetraploid has 4 copies of their genome, and we look at a small location on their genome. This individual has 3 Cs and 1 G at the SNP, this location where there's a variation. And so the dosage of C for this individual is 3. But if an individual had 2 Cs, that dosage would be 2. So the possible dosages for an individual is from 0, 1, 2, 3, or 4 for a tetraploid individual. For higher ploides like 6 or 8, there would be more dosages possible. So the naive thing to do for Hardy-Weinberg equilibrium tests in polyploids would be to compare the counts for individuals with each dosage against the binomial distribution. So let xi be the dosage of C for individual I, then say, all right, Hardy-Weinberg equilibrium is fulfilled if xi is binomially distributed with size k, where k is diploidy, the number of copies of the genome an individual has, and success probability PC. And then, so if this were the case, and this is what Hardy-Weinberg equilibrium meant for polyploids, then you could just very simply extend all of this machinery from diploids to polyploids. You could use chi-squared tests, exact tests. You could use all of that machinery and expand it, and it would be relatively straightforward. So the classical approach is, this was done long time ago, back in like the 50s. But not too many people have extended the other exact tests or accounting for structure to polyploids. But in principle, you could, right? This would be relatively straightforward. The issue is that the binomial distribution isn't really representative of equilibrium in polyploids. So in autopolyploids, there's a phenomenon in meiosis called double reduction, which creates a dependency between the alleles. And this dependency alters those binomial proportions. And so if you just apply these naive methods to that assume a binomial distribution, you would have a bias test. So you would have a test that would have a higher type 1 error than what you're controlling for. So let's talk about meiosis in autopolyploids and talk about how this can cause this dependency. So meiosis is the process by which gametes are produced from an individual. You create a gamete by sampling half of an individual's genome. And then new individuals are created from those gametes by combining with other gametes. So for a diploid individual at a locus where there's a GG, a CG, or a CC, so for a GG individual, they're going to create just G loci, G gametes. For a CG individual, they're going to create a G gamete with probability 1/2 and a C gamete with probability 1/2. And for a CC individual, they'll just create C gametes. So this is what happens in diploids. In polyploids, you create gametes with half the number of copies of your genome. So for a tetraploid, if you have 4 copies of your genome, then you're going to create a gamete with 2 copies of its genome. And then later during sexual reproduction, these gametes would combine to create a new individual with 4 copies. So if there were independent assortment, so if all of these alleles were chosen independently to be placed into the gamete, then you would expect the dosage for the gamete to follow a hypergeometric distribution. To see this, you can think about this in terms of the ball and urn model. So if an individual is an urn and they have CC to GG, and the C allele is a white ball and the G allele is a black ball, then there are 2 C's in this individual, in this urn. There are 2 G's, 2 black balls in this urn. And to get the dosage of a gamete, you randomly choose 2 balls and you count the number of white balls, the number of C's. And so this would be a hypergeometric distribution. So if there were independent assortment, you can show that this hypergeometric distribution would eventually result in those binomial proportions at Hardy-Weinberg equilibrium. The issue is that in autopolyploids, there's a bias for homozygous gametes. So gametes that have 2 C's or 2 G's compared to heterozygous gametes. So gametes that have just 1 C or 1 G. And the amount of this bias is called the double reduction rate. So this bias makes it so that at Hardy-Weinberg equilibrium, you no longer have those binomial proportions. So let's look at what this bias looks like. So here I have different facets, and these facets are different dosages for an individual. So an individual with 0 C's, 1 C, 2 C, 3 C's, or 4 C's. And then on the x-axis, I have the resulting gamete dosages that that individual would produce. And on the y-axis, I have the probability of an individual with that dosage producing a gamete with a given dosage. So for example, right here, this orange bar, this means that the probability of a gamete with a dosage of 1 being produced by an individual with a dosage of 3 would be 0.5. Now, the different colors are the different models for meiosis. So orange would be that hypergeometric distribution that you would expect under independent assortment. And then black would be under a model where there's double reduction. So this process in meiosis that is specific to autopolyploids. Now, when there's double reduction, you have lower probabilities of there being a heterozygous individual. So a dosage of 1 for the gamete. Those probabilities are smaller, and there's a higher probability of homozygous genotypes. Indeed, in cases where there's 0 probability under the hypergeometric model, there's actually a non-zero probability of having some gametes of that dosage under when there's double reduction. So these altered frequencies under meiosis can actually ultimately result in altered frequencies under the binomial distribution when you have equilibrium. So this is a little cartoon for meiosis. This would upset my biologist colleagues because it's too simple, but for me this is good enough. So each of these colors are a different genome of an individual, right? So this is a tetraploid individual, and they have 4 copies of their genome. So for the meiosis, the first step is that all of these copies make a copy of themselves. These are called sister chromatids. So this blue creates a copy, this pink creates a copy, green creates a copy, red creates a copy. Now, these copies, these sister chromatids, segregate together during one part of the meiosis. So here, green migrates with green, blue migrates with blue, pink migrates with pink, red migrates with red. And then which colors migrate together, this is a random phenomenon. And then after this, the sister chromatids segregate into different gametes. So one green goes here, the other goes here. One blue goes here, one blue goes here. So in this way, a single, an individual with 4 copies of their genome can create 4 different gametes. What double reduction occurs is when there's crossover between the different copies of the genome. So crossover is where there are breaks on the genome, and then they interchange, they swap parts of the genome. So here, part of this blue genome swaps with part of this green genome. And if you have alleles on this part of the genome, then it can result in altered segregation patterns. So here, again, these blues, the primarily blues segregate together, the primary greens segregate together, and then this could possibly happen where 2 blues for this section of the genome migrate together. So even though there's only 1 blue in the individual, there could be sections of the genome that contain 2 blues in the gamete. So if, for example, if this individual has a dosage of 1 and the only C is on the blue genome at this location where there's a dot, then they could produce individuals with 2 Cs. This would not be possible in diploids, but in polyploids and autopolyploids, this is a possible phenomenon. So why this matters to us is that this makes CCs and GGs more likely and CGs less likely. And because of this, there's this dependency in the population between the different alleles. And this dependence distorts these segregation, these binomial frequencies. So in order to quantify what this distortion looks like, I have on these different facets, different dosages that are possible. A dosage of 0, 1, 2, 3, and 4. And then on the x-axis, I have different allele frequencies. So how likely each allele is in the population. And then on the y-axis, I have the genotype frequencies when there's double reduction minus the genotype frequencies under the binomial model. So this black curve, whenever it's above the red line, that means that when there's double reduction, this genotype, in this case, the genotype of 0, is overrepresented compared to the binomial distribution. When it's below the red line, then this genotype, in this case, a genotype of 1, is underrepresented in the binomial in the double reduction model compared to the binomial. So what you can see from here is that homozygous genotypes, where an individual has all the same copy of an allele, is overrepresented when there's double reduction compared to the binomial distribution. But for other cases, it's more complex what's going on. But the issue is that these frequencies are going to be different than the binomial distribution. And because they're different from the binomial distribution, methods that assume a binomial distribution are going to behave poorly. So here I ran a likelihood ratio test assuming a binomial distribution. I measured its type 1 error controlling at the 0.05 level at different levels of double reduction. These are all realistic levels of double reduction. And only when there's no double reduction or very little double reduction in meiosis do the naive methods work. And in fact, when there's a lot of double reduction, the naive method completely fails. It's completely unable to control for type 1 error. Yeah. So I'm going to talk about briefly 2 methods that I came up with to test for equilibrium when there's double reduction in the population. So there's a very useful paper by Kang Huang and his colleagues, where given an allele frequency and a double reduction rate, which we'll call alpha, you can come up with what the equilibrium frequencies are for different ploidies. They did it for 4, 6, 8, and 10. So if you have these equilibrium frequencies at a given allele frequency and a given double reduction rate, then in principle it would be easy to develop likelihood approaches to test for equilibrium. And so I did this, but the issue is that these equilibrium frequencies are really complicated. They're not very useful for generalizations. So they only did it for 4, 6, 8, and 10. And I think this is for computational reasons. So how they came up with these frequencies, these genotype frequencies at equilibrium, is that they use symbolic algebra software like Mathematica or Maple or something like that. They set up a system of equations and then they solve the system of equations, and it gives you this complicated formula. Now, when I did this, I was able to do it for 4, 6, and 8, but not for 10. My desktop was not able to do it. And I suspect that they weren't able to do it for 12 and higher. And then it's not, because of how complicated they are, it's not as easy to generalize to other scenarios like multi-allelic loci or other forms of meiosis than just those that include double reduction or mixed ploidy species like those that are mixed between diploid and polyploid. It would be much harder to generalize this approach. So just to give you an idea of what these frequencies look like, on the next slide, after this one, I'm going to give you the gamete frequency at equilibrium for a dosage of 3. So the probability that a randomly selected gamete would have a dosage of 3 from a decaploid species, so a species that has a dosage of 10, possible dosage of 10, so 10 copies of their genome. And it'll be in terms of the allele frequency, and then 2 parameters for the double reduction rate. So for tetraploids and hexaploids, there's only 1 double reduction parameter, but for higher ploidies, there's more than 1. So it's in terms of 2 double reduction rates. So this formula that I'm about to give you, it's just for the probability of 1 gamete dosage at a dosage of 3. Now, if you want the probability of a genotype dosage, then you would need to perform a convolution of the gamete frequencies. And so the actual, so I'll just show you the gamete frequency at a dosage of 3. So here it is. And so if you do genotype frequencies, you're going to be doing a convolution of a lot of things that look like this. And so the actual genotype frequencies, if you wrote them out, they would span pages of lines of math, just like this. So it's very complicated. In the computer, you can put this into a computer, and then it's not so complicated for the computer. But if you want to develop generalizations, then it's very, very complicated. It would be hard to do that because there's very little intuition that you can gain just by looking at this equation. So how would you do likelihood methods, likelihood ratio tests for Hardy-Weinberg equilibrium in autopolyploids using these frequencies? Let Y be the number of individuals that have the different genotypes. So 0, 1, up to K. And suppose Q contains the genotype frequency. So the proportion of individuals that have a dosage of 0, dosage of 1, dosage of 2, up to K. Let R be a vector of gamete dosage frequencies. So it's this is a K over 2 plus 1 vector, where each element is the probability of randomly selecting a gamete with a given dosage. And these are those complicated formulas from Huang, 2019. So then if equilibrium were fulfilled, then Q would be a discrete linear convolution of these gamete frequencies. And the reason why is that genotypes are sums of gametes. And so if you have the sum of two independent random variables, then you're going to have a convolution. So this is the reason why. So the likelihood would be a multinomial distribution with size n for the number of individuals and category probabilities q, where this q is either unconstrained except to sum to 1, or where it's constrained to be this if equilibrium were fulfilled. So we obtain p hat and alpha hat by maximum likelihood. We obtain the empirical genotype frequencies for the alternative model of no equilibrium. We set up a likelihood ratio test statistic, and we compare it to the appropriate chi-squared distribution. And so this is how you would do a likelihood ratio test for equilibrium in autopolyploids. But again, this would be very difficult to generalize to other higher ploidies, to other forms of meiosis, to more complicated population genetic models. So my goal was to develop a conceptually simpler method that would hopefully be easier to generalize. All right. So in order to do this, let's let PR, so G given G1, G2, and theta, be the probability of producing a gamete with dosage lowercase g, or not producing gamete, sorry, producing an offspring with lowercase g given parental genotypes cap G1 and cap G2. So this is, this depends on the form of the meiotic model and the parameters of that model. And so we'll just denote all of the parameters of that model by theta. In the case of this paper, we'll let theta be the double reduction rate. But if you have other forms of meiosis that you want to model, like for odd ploidy or mixed ploidy species, then theta could potentially be different. All right, so PRG given G1, G2, and theta is the probability of producing an offspring with lowercase g dosage given parents with capital G1, capital G2 dosages. Let XI, XJ, and XL be the genotypes for individuals IJ and L. So my idea is to compare the genotype for individual I to the probability that individual I would have that genotype if individual I's parents were individuals J and L. So just choose two individuals, J and L, to act as the parents to individual I, and then compare the probability of individual I having a given genotype to whether or not individual I actually has that genotype. So this one is an indicator function. So 1 if individual I has genotype G and 0 otherwise, and we're comparing it to the probability of genotype G if individual I's parents were J and L. So let's look at the statistic. Now, at equilibrium, the genotypes for individuals J and L would be identically distributed to the genotypes of individual I. Just by the definition of equilibrium, if you were to randomly select individuals J and L, they would be identically distributed to the genotypes of the parental generation, because the genotypes would not be expected to change from generation to generation at equilibrium. So because of this, it's pretty easy to show that this statistic has expectation 0. So the expected value of the indicator is just the probability of that genotype. And then the expected value of this probability, you're just integrating over the parental genotypes. And if it's identically distributed to the actual parents, then it's the same thing as this probability. So it's pretty easy to show that this statistic has expectation 0. But only at equilibrium. If it's not equilibrium, then it would not have this expectation being 0 because the parental genotypes would be differently distributed than individuals I and J and L. So we're just looking at 3 individuals here, but those 3 individuals were arbitrary. We could just look at all triples of individuals and then average the statistic. And the statistic would still have expectation 0. So this is what this is doing. It's creating a statistic called HG of theta, which is the average of all of those HG statistics that each have expectation 0. So this is a U statistic. This is what we call a U statistic in the literature, because we're averaging over all triples of individuals, basically. So this is actually not just one U statistic, but this represents K+1 U statistic for each of the possible genotypes that we could be comparing. So genotype 0, 1, up to K. So I'm going to put all of those K+1 U statistics into a vector, H of theta, where each of these H's have expectation 0. Now, due to sampling variability, these HGs will not actually equal 0. They have expectation 0, but they'll deviate from 0 just because of sampling variability. But because they should equal 0 in expectation, we can, one estimation strategy for estimating this theta, this parameter of meiosis, would be to minimize a norm of this H of theta. So that's what this is doing. It's minimizing a norm of this H of theta, where omega is some positive semi-definite matrix. So this is an estimate. So if theta is the double reduction rate, this is how we would estimate the double reduction rate using this new U-statistic approach. And you can show, actually, that for carefully chosen values of omega, that this minimized norm follows an asymptotic chi-squared distribution, but only under equilibrium. So because this minimized norm follows an asymptotic chi-squared distribution, but only under equilibrium, you can compare it to that chi-squared distribution to obtain a p-value against the null of equilibrium. And so this is the new approach. So just to summarize, we came up with a statistic that has expectation 0 that uses 3 individuals. We averaged that statistic over all triples of individuals, so it still has expectation 0. This is actually k+1 statistics that we put into a vector. Now, it'll differ from 0 because of sampling variability, and so we minimize a norm of the statistic in order to estimate the double reduction rate. And this minimized norm follows an asymptotic chi-squared distribution that we can use in order to test against the null of equilibrium. So this is actually connected to a lot of literature. Estimators where you minimize a norm of a vector view statistics are sometimes called U-process minimizers or MM estimators. And there's a little bit of literature there. I spent hours trying to find something similar to this, and I wasn't able to, and so I had to consult a friend of mine who's an expert in U statistics on whether he's seen it before. So it's very little literature, but it is out there, and it's really nice because it gives you consistency guarantees of your estimator. So as you collect a large enough sample size, you get more and more accurate estimate of the double reduction rate. And then this idea of minimizing the norm and comparing it to a chi-squared distribution, this is actually connected to the generalized method of moments literature. So you can think about this chi-squared test as an application of the Sargan-Hansen J-test from econometrics, which I think is a kind of cool connection. So the way that I described it, you would think that it would take n cubed time in order to calculate the statistic, but it turns out that there's an equivalent representation of the statistic in terms of just the genotype frequencies. So again, Q is the vector of genotype frequencies, the proportion of individuals that have each genotype. Let Q hat be the sample genotype frequencies. And then let's define this F to be a function that updates the genotype frequencies from one generation to the next when there's random mating and some meiotic process parameterized by a parameter theta. So F is, so if Q is contains the genotype frequencies of the current generation, then one generation later, those genotype frequencies will be F of Q. So one can show that this H vector that contains statistics that have expectation 0 is just Q hat minus F of Q hat, which is really nice because at equilibrium, you would expect Q to equal F of Q. And so that means that Q hat would be approximately equal to F of Q hat in expectation 0 specifically. So this is really nice, but you need to use this U statistic formulation before by considering these triples of individuals in order to come up with the appropriate asymptotics and derive the asymptotic distribution of that norm. So this is really nice for computation, but in order to prove those results, you need to use the U statistic approach. I will skip going through an example implementation of this. And I don't think I have time for talking about the simulations, but I ran some simulations. The only thing that I'll talk about, so these are estimates of the double reduction rate for different methods. There's only one method that I could find that claims to estimate the double reduction rate in natural polyploid populations. But they do it wrong. They have an incorrect model for double reduction for the equilibrium frequency of the genotypes given a double reduction rate. So this paper is from Zhang and company from just this year, 2021. And here I have the MLE and the U statistic approach, where this red dashed line is the true estimate, the true value of alpha, and as you know, they're unbiased. The MLE and the Ustatistic approach. Maybe the Ustatistic approach is a little bit better, but they're both performing approximately the same. And then, but this Zhang paper, their method, it's highly biased. So that's the only thing I'll say, is that our approach appears to be biased. The Zhang method is highly biased. But even for a very large sample size, like 1,000 is a huge sample size for polyploid studies. There's still a high amount of variability in these estimates. So it's very difficult to estimate the double reduction rate given a single biallelic locus. Okay. So, okay. Yeah, and then it appears that this, you know, the MLE and the Ustatistic approach are asymptotically similar. Another thing to note from these simulations. So to conclude, we used the equilibrium frequencies from Kang Huang and company from that nice paper to develop likelihood methods to estimate double reduction given equilibrium and to test for equilibrium while accounting for double reduction. They didn't do that in their paper, but we used their results in order to do it here. But in order to develop something that was conceptually more simple and possible to generalize, we developed this new Ustatistic approach. It's up on CRAN for the HWEP package, WEP. So go check that out. And then for this other paper, we do some other things in this paper. So I discussed the differences between random mating and equilibrium in some of the literature. This has been confused, and so I talk about it more explicitly. I implement tests for random mating, and then, you know, genotype uncertainty is a big issue, specifically for polyploid studies, where you're not sure about what genotype each individual has. And so in order to account for genotype uncertainty and test for equilibrium in the presence of genotype uncertainty, I developed bootstrap approaches. So go check that out. So thank you to my new funding, and I will take any questions.
I think we should thank David for an outstanding presentation. And I'd like to apologize, David. I got you the most powerful computer that I thought I could at the time, and I'm sorry you couldn't run the simulations for 10, but there is some capability to doing shared time on a you know, a so-called, I guess you would still call it a supercomputer. So John and Betty also know about that. So you talk when next time I'm on campus, I'll talk with you about that. So
no, I have access to Zorro. I didn't try it on Zorro. Okay. I could have tried it on Zorro, but I didn't. Yeah.
But that was an outstanding presentation. Any questions?
So if no one else does, got a couple. So is there any sort of max ploidy? I mean, is there any biological limit?
Oh, yeah, that's a great question.
What's K?
So most polyploids are 4 or 6.
Yeah.
But there are some polyploids with way higher, like 100, right?
Really?
So there are some, but it's you know, they're not well studied organisms. So there are some, but most are 4 or 6.
These are single cell, or are they?
No, so I don't know exactly. I would need to look it up. I know Mike Barker's lab would know more. They study like the distribution of ploidy across the world, so they would know more. So I would have to look it up to know exactly what's going on.
Yeah, I'd be curious to know the one that has 100. That's pretty well.
Yeah, I'm pretty sure there's something like that if I remember right. But you know, most have 4 or 6.
Dr. Gill?
Is there?
So I had I guess probably a naive biology question. Everything I learned about genetics was in a medical school, so we only care about diploids. But I understand that like plant mating, which I'm sure is the wrong term, can be very complicated in terms of the combinations and the crossovers. And so is this frontier of complications you have in front of you after this paper?
Yeah, that's a great question. So so dealing with mixed ploidy species, I think is really interesting to me. So there are a lot of species where diploids and tetraploids live in the same area, and they mate between each other. And so being able to study jointly whether or not they're in equilibrium would be interesting, as opposed to studying each diploid and tetraploid separately if they're in equilibrium. So that would be the next step of this project. Yeah.
Okay, thanks.
So John, I'm sorry, your second question? Dr. Nolan?
Well, there are a couple, but I'll stick to one. So is there any understanding in this polyploidy if there's sort of a trend? Do we start from diploids and go to tetroids and et cetera, or
so how do
cycles throughout history where there were a lot of polyploids and they sort of faded out? Or
that's a great question, and again that's something that I'll be talking about as not an expert, just something that I've seen from other presentations. But my understanding is that in our evolutionary past, there has been multiple polyploidization events. So it's actually an important process in evolution in general. So there's a lot of ancestral polyploidy. And the way that polyploids form, there's multiple ways. So either there can be a whole genome duplication event, in which offspring have just doubled their genome, or there can be a split off evolutionarily, and then a hybridization event. And then in this hybridization event, that's how the genome duplication occurs. So those are the different processes by which polyploidization occurs. But again, I'm not an expert in that area. I always depend on my biologist friends to help me out there.
Is a trisomy mutation sort of a random?
Yeah, so triploid individuals are less common. So usually even ploid is more common than odd ploid, because during meiosis, you have to split it into two. And so if you have a triploid individual, that split can create problems with fertility. And so, triploid sexual reproduction that does exist, but it's very rare. It's mostly those individuals tend to be more sterile.
Do we have any other questions for David? I think we should go ahead.
I just have one or two, but I'll wait till I'll give everybody else a chance to leave and congratulate you.
All right. Maybe on that note, I think we should thank David for a really outstanding presentation. And we wish him continued success with his work, and we need to make sure we get him all the computing power he needs. So with that, thank you, David.
Thank you so much.
It's an excellent talk.
Thank you.
Excellent
Good talk.
Thank you.